It is my great pleasure to introduce Renee Gallagher. Renee is a Senior Specialist in Genetic Diseases at Illumina. Her presentation is about sequencing and genomics technology in healthcare. Welcome, Renee, and we look forward to your presentation. Thank you.
So, thank you. Today I'll be talking a bit about Genomics and Sequencing 101 and Genomics Technology in Healthcare Review. I'm a Senior Specialist in Genetic Disease and I cover Australia and New Zealand. So, but I have been working in the laboratory setting for about 15 years. I've worked in cytogenetics and molecular biology and just recently moved into industry where I get to continue to do a lot more of what I was doing, but on a much greater scale. So at Illumina, I'll just make sure that my slides are moving along. 1 second. So at Illumina, our mission is unlocking the power of the genome to improve human health. And I think you'll find it's quite difficult to find someone that works for Illumina that doesn't personally align with this mission as well. We are all very passionate about improving human health by genomics and implementing genomics into routine healthcare. And one of the many ways to do this is by leveraging our technology. So we have workflows from sample all the way through to answer, and that's laboratory workflows and bioinformatics solutions. And one of the things we do is we continue to evolve our technology and applications to make sure that we have the ability to help the adoption of genomics in the clinic and drive a lot more diagnoses as we need to. We have 6 major areas that we focus on. We have oncology, reproductive health, microbiology, agriculture, molecular and cell biology. But the area that is closest to our hearts, especially tonight, is rare and undiagnosed genetic diseases. And we have everything from sample preparation, microarrays and sequencing through to informatics solutions that empower researchers and clinicians across the globe to find the answers that they seek in rare disease research and within the clinic as well. So that's a bit about who we are. I know Rihanna gave us a beautiful overview last month about genomics, but I'll do the same to make sure we all have the same baseline going forth into the rest of this webinar. So I just wanted to give you some fun facts about DNA. We know that DNA encodes for the building blocks of life, but I just wanted to take a step back and have a look at the human body as a whole. So there are about 37 trillion cells in the human body, and within each cell there are different organelles and other macromolecules, like we have the mitochondria, which is the powerhouse of the cell, and we also have the nucleus, which houses most of the genomics materials. So we've got chromosomes. We have 23 pairs of chromosomes, so 22 autosomes and 1 pair of sex chromosomes. And if you were to pull apart those chromosomes, they would work out to be about 2 meters in length. If you further look at that DNA, you'll see that there are about 3.2 billion nucleotides, and they're made up of adenine, guanine, cytosine, and thymine. And one of the really important things about DNA is that there's less than 1% difference between individuals, which I find rather fascinating. So we're just going to do a deeper dive into what are chromosomes. So DNA is packaged into thread-like structures called chromosomes, and each chromosome is made up of DNA tightly coiled around proteins called histones that help support its structure. We have 23 pairs of chromosomes, so 22 autosome pairs, and 1 sex chromosome pair, so XX female, XY male. And the study of these chromosomes is called cytogenetics. We have a very typical male karyotype from blood in the image on the right, and the resolution of blood karyotyping is usually about 5 to 10 million bases. Now, to put that into perspective, chromosome 21 is about 47 million bases in size. Now, one of the most commonly known chromosomal abnormalities that we know is chromosome trisomy 21, so Down syndrome. What is a gene? So there are 2 types of DNA. We have coding DNA and about 1% of the human genome contains protein coding genes. And the protein coding sections of genes are called exons. And this will be important to remember later when I'm discussing the various types of sequencing. So a gene has exons and introns, and there are about 20,000 protein coding genes within the human genome. Genes contain the basic level of information to make molecules called proteins, and the activity of genes are tightly controlled, and loss of this control can lead to diseases like cancer or other genetic diseases. Now, non-coding DNA, about 99% of the human genome is not protein coding, and it was originally thought to be junk DNA with no purpose. There are many types of DNA regulatory elements that we now know about, and these include promoters, which are recognition sites for gene activity, enhancers, which help to activate a gene, silencers, which repress gene activity, and insulators, which control gene activity either through enhancer blocking or preventing access, so structurally. We also know about the specialized RNA molecules now, so we have things like transfer RNA molecules, ribosomal RNA molecules, microRNAs, and long non-coding RNA. And there are other structural and regulatory elements as well. So let's do a deeper dive. What is DNA? So deoxyribonucleic acid, I think you can understand why most scientists talk about it as DNA, carries the genetic instructions for the development, functioning, growth, and reproduction of all known organisms. Information stored in DNA is stored as a code made up of 4 chemical bases: adenine, guanine, cytosine, and thymine. Human DNA consists of about 3 billion bases, and more than 99% of those bases are the same in all people. The order of the sequence of these bases determines the information available for the building and functioning of an organism. But what's the structure of DNA like? So DNA bases pair up with each other to form units called base pairs. And again, this is a really important concept for when I talk about sequencing in a moment. So adenine will pair with thymine, and guanine will pair with cytosine. And each base is also attached to a sugar molecule and a phosphate molecule, and that is what creates a nucleotide. And the nucleotides are arranged in 2 long strands that form a spiral, so the double helix that we understand. Now, there are 2 processes that are really important to create a protein. So in transcription, the double helix separates to provide access to the DNA code. The RNA molecules bind to their complementary DNA partner and form the message RNA molecule. The message RNA molecule is transported out of the cell nucleus and attaches to a ribosome. So within the ribosome, the mRNA binds with the complementary transfer RNA molecules, and then each tRNA molecule recognizes a group of 3 bases called a codon, which then codes for an amino acid. And this is the basic component of protein molecules. The adjacent amino acids are linked together to form a protein. Now, what is the impact of an error in the genetic code? So there are about 4 million differences in the genetic code between individuals. And this variation could be a single letter difference or could include large sections of DNA. Depending on the location, it could have little or no impact, or others could have severe consequences. Genetic code variation could result in a non-functioning or truncated protein, potentially causing a genetic disease. Regulatory elements may not function correctly, and the tightly controlled nature of the mechanisms within the cell or organisms could be destabilized. So if we take a look at the figure on the left, this is our string of DNA and the proteins and the amino acids and the protein it codes for. Now, a singular G to T could cause a change in the protein, or a G to a T elsewhere could cause termination of the protein. So it will change the entire function or just allow that protein to be available for any of the cellular processes required. So now I've covered this, I want to talk about some sequencing fundamentals. Sequencing is basically the ability to read the genetic code. To be able to read the sequence of DNA, it requires the ability to distinguish between the different nucleotides. And there are various methods available. Some recognize the size and shape, but the majority use pre-labeled fluorescent nucleotides that only bind to their complementary partner. So remember A and T and G and C. And you can see here that there are 4 colors denoting the different bases. So T is blue, G is green, A is red, and C is yellow. In 1977, Sanger-based electrophoresis was invented, and this enabled 1 read per capillary and up to 96 capillaries per run. So this enabled 1.2 million bases of data generation per day per machine. And this was singular read technology. Now in 2007, Illumina released something called next generation sequencing using cyclic reads. It enabled, and today it enables about 4 million to 20 billion reads per flow cell and about 1,500 gigabases per day. And it's also important to note that this is now what we call massively parallel sequencing. So there are multiple DNA fragments or reads that can be sequenced at the same time. So how does it work? I'll talk a bit more in detail in a minute. But essentially, it uses sequential images to create the DNA sequence reads. So each image on this slide is of the same part of the flow cell. And each of these fluorescent dots is a DNA molecule. Now, if we were to take a look at the top left, we can see that in the first cycle, there was a green color detected, and this denotes a G. In the second read, a blue nucleotide was detected, so this was a T. And you can start to see as each cycle occurs, you can build out these reads. So essentially, that top left DNA molecule can read as GTA, GCA, TCG. Now the bottom right, you can see that it's a long string of Gs, a singular T followed by a G. And the cyclic read technology enables that really accurate base calling that can read through these long stretches of the same base, which is really important when it comes to the human genome. So what do our machines look like? So we have basically sequencing for every scale. So we have our baby, which is like our iSeq 100, and that can do 1.2 gigabases at a time, all the way through to our NovaSeq 6000, which is our more production scale sequencer, and that can do about 6,000 gigabases per run. And this is where we see most of our genomes and exomes be performed. Most labs, when they're first doing sequencing, will either have a MiSeq or a NextSeq in their laboratories. And in fact, the MiSeq was the very first machine I worked on from Illumina many, many years ago. And it's a fabulous machine, and that lab is still running it. So the evolution of technology is allowing this decreasing cost of sequencing. So to put this into perspective again, the first draft human genome cost $300 million to create, but it still needed refinement. And so over the course of the Human Genome Project, the actual human genome cost $2.7 billion USD to sequence, and this took more than 10 years. Now, today's human genome sequencing, it can cost as little as $1,000 US. Takes 24 hours, and this reduction is 100,000-fold. But it's also kind of cool to put it into perspective. It's not only human genome sequencing that occurs. There are animals, plants, microbes, and then there are different types of sequencing. There's DNA sequencing, RNA sequencing, epigenetic studies, and much, much more. So I'm going to talk now about the sequencing workflow. And I think this is where you'll find it most interesting. So I'm going to talk about the example life cycle of a clinical sample. So step one, the patient is cared for by a specialist and identified for genomic testing due to presenting features. This could be multiple congenital abnormalities, laboratory abnormalities, family history, etc. or you yourself are advocating for your child to have more genomic testing. The option of genomic testing is presented to the parents by the clinicians or the genetic counsellor. Consent is obtained, and then blood samples or saliva samples are obtained on the child and sent to the laboratory. The signed test requisition, consent forms, and a summary of patient's clinical presentation are provided by the ordering MD. And I think this right here is one of the most important things that I can say to you as a parent, is to ensure that as much information is sent to the laboratory as possible by your doctor when they're requesting genomic testing. The more information the laboratory has, the more information that the curators or the interpreters at the other end of sequencing have to help determine a diagnosis. When the sample is received by the laboratory, it's accessioned and then loaded onto the sequencer. The sample is held if any of the required information is missing. So it's really imperative that everything is in order, the consent, the request forms, and the phenotypic information is all there ready for the laboratory so the sample can be processed straight away. Once the sample is sequenced and meets quality control metrics, it is then sent for processing by Bioinformatics. And raw sequencing data is processed through the Bioinformatics pipeline, and there are guidelines, so standards. So in Australia, it's NATA, in New Zealand, it's the INZ, but there are global standards that every country has to meet for clinical reporting. When the sample meets the quality metrics, it's then sent to the interpretation team. And the clinical interpretation team inputs the sequencing data into filtering software tool. And they often utilize the information provided by the doctor about the child's phenotype to help identify candidate variants and genes for curation and further interpretation. The clinical report is then delivered to the ordering clinician, and the results are then disclosed between the clinician and the family. So this is the common workflow for all applications from the laboratory point of view. The sample is extracted and purified for the next step in the process. And then in library preparation, this is effectively preparing the sample for sequencing by adding special adapters that are recognized by the sequencer. In sequencing, this reads the sequence of the sample and creates the raw data. And then in analysis, the analysis of the sequencing data has 3 stages: primary and secondary analysis, followed by interpretation. So to take a deeper dive into library preparation, we have our genomic DNA, and if it's from blood or saliva, it tends to be what we call high molecular weight, so really intact, long stretches of DNA. So to enable sequencing, we need to fragment them. So we effectively chop up the DNA into smaller pieces. And this is what is denoted by the DNA insert, so the grey bar here. So there are 3 things that need to be added to the DNA insert. So for clustering, libraries must have this P5 and P7 binding region on either end of the library. For sequencing, libraries must have the sequencing primer binding regions added as well. And then for pooling a sample, so to economize sequencing and keep the cost of sequencing down, we add unique identifiers to each sample. And this allows us to then pool the samples and get the most bang for your buck. So what does this look like further? What type of library preparation methodologies can we talk about? So we have targeted sequencing, which are more like your gene panels, and it's effectively a defined subset of genes, and they're usually disease specific. And the laboratory tends to design these because they assay only what they want to report. There's also Whole-Exome sequencing, which effectively is the hybridization capture of the exons, and so the coding part of the DNA. And the reporting is generally restricted to the coding and splice site sequences, so the exons themselves or just outside them. Whereas in whole genome sequencing is the most comprehensive representation, as it's the entire genome. The reporting can consider all variants, and there's increased sensitivity versus the exome. And of course, it has the highest diagnostic yield because you're assaying the entire genome. So to just further talk about the differences between whole genome sequencing and whole exome sequencing, I just want to bring your attention back to the gene structure. So we have the exons and the introns, etc. So we have our sample, the genomic DNA is extracted, then we do our fragmentation to chop it up, and essentially we are left with a DNA sample that has both introns and exons all within it. Now, in whole genome sequencing, this is effectively put straight onto the sequencer after adapters have been added to it, and then after sequencing is sent through the bioinformatics pipeline to report. Now, in whole exome sequencing, it requires a few extra steps. So there are complementary sequences of DNA that effectively bind the exons out of the whole genomic DNA pool. And then after these are after the non-bound DNA is washed away, we're left with what we call the captured exons. Because these represent a much smaller amount of DNA now, we have to do a process called amplification to increase the amount of DNA so it can go on the sequencer. Now, after sequencing, it can go through a very similar analysis and interpretation bioinformatics pipeline to report. So you can see that it's very similar, but there are just a few different nuances within the laboratory processing. Now the sequencing itself. This is very cool. So there are two steps that need to occur for Illumina sequencing. There's something called cluster generation and then sequencing by synthesis. In cluster generation, the prepared DNA or the prepared library is flushed across the flow cell. And on each flow cell, there are the complementary partners to the P5 and the P7 adapters that I spoke about earlier. And so the DNA inserts attach to these flow cells via these complementary oligos attached to the glass. And essentially what happens is multiple copies are made. So clusters are generated, so little bunches of the same DNA molecule. And this just enables the detection of the incorporated nucleotide through the sequencing to be better detected. Then once this occurs, sequencing by synthesis can happen. So what happens here is that all four fluorescently labeled nucleotides are added to the flow cell. Now there is natural competition between the A's and T's and the G's and C's to bind to their complementary partner. So this is how we ensure the high accuracy of sequencing by synthesis in Illumina technology. So the base is added to the DNA insert, and in this cartoon, it's green, so it's a T. I'll just get, yeah, perfect. Get the little thing happening. All right, so then in every cluster, when the first base is added, it'll turn that entire cluster the color of the base. So again, like I said, in the first image, it's a T because it turned it green. Now, once the first base has been imaged and determined, the next cycle can occur. So all four labeled nucleotides are then flushed across the flow cell and the second base is added. In this case, the cluster was determined to be blue, so therefore a G. Now that's the sequencing technology, but what does it physically look like? So the NovaSeq 6000 essentially just looks like a really big washing machine. There are 4 different types of flow cells that can be utilized for it. There's the S prime, the S1, the S2, and the S4. So, and these just determine like how much real estate is available. So the S prime flow cell is our smallest flow cell, and the S4 flow cell is the largest. So to put that into perspective for how many patients or samples can be run, we look at exomes. It's about 40 exome samples that can be run on an S prime and only 4 genomes per run. Now, if we contrast that to the S4 flow cell, it's about 500 exomes per run, or about 48 genomes. So you can see that there is capacity to sequence what you require, and if it's a rapid turnaround that's required, all the way through to quite a large number of samples, and is still within a decent time frame, so that can be done in less than 48 hours. So when it comes to the laboratory work, that's all relatively easy these days. It's been refined to small art in that sense. It can be automated. It doesn't require a lot of hands-on these days. It can be done very quickly. Where the time is spent now is looking at bioinformatics and how to interpret the sequencing data. So I just want to touch on a bit more about the bioinformatics, and then we'll talk about variant interpretation. So the typical bioinformatics workflow has the three stages: the primary analysis, secondary analysis, or the tertiary analysis, otherwise known as the interpretation. So in the primary analysis, this runs in parallel to the sequencing. It uses the images to provide base calls of the reads. So again, we're getting very familiar with these images where we can start to pull out what the sequences are per cycle. So the G, T, A, G, C, etc. The secondary analysis happens after the sequencing is finished, and this maps the reads to the reference genome. So this is what we call alignment. It analyzes the mapped reads for any differences compared to the reference genome, and this is what's called variant calling. The tertiary analysis or interpretation, this is what leads to the knowledge and insights into basic biology, and also the potential causes of genetic disease. This enables us to look at whether or not there's possible treatment or prevention that can be utilised as part of that diagnosis. But I want to take a deeper dive into bioinformatics so that everyone has a decent understanding about what's happening with the data. So like I said, the primary analysis occurs in real time, essentially. It converts genomic signals to nucleotide positions and ordering. Demultiplexing of the samples re-identifies the sample from individual reads. And I'll talk about that a bit more in the next slide, but if you remember those unique indexes that essentially allow us to identify each individual sample before we pull. And the outputs are usually FASTQ files. So this is a cartoon-like image of what demultiplexing looks like. So if all of these are the individual reads, and those little coloured blocks are the individual indices used per sample, we can start to look at this bioinformatically and determine what samples belong in each sample bin. So each read is assigned to the sample, and this assignment allows the FASTQ files to be generated for a sample. And this, again, is a way for us to economize the sequencing. In secondary analysis, this is the sequence alignment. So we have a reference sequence, and this is denoted in the teal. We have our sequence reads, and examples of this are in the red. And then we have the consensus. So this is where we have a look and see whether or not we can call what the patient sequence looks like. So the alignment of raw sequencing to the data reference occurs. The identified changes from reference, or the variant calling, happens. And then this is done to perform sequence variations from the reference. And these sequencing variations can include such things as single nucleotide variants and small insertions or deletions, copy number changes, or structural changes. And the outputs here are usually BAM and BCF files, or variant call format files. So the types of variants that can be determined from next generation sequencing include single nucleotide variations, so a single base change. And you can usually see these in these things called reads, and that's what these are here, just the aligned reads to the sequence. There's translocations, so a swapping of genetic material amongst the chromosomes. Copy number variation, so if there's a normal copy number of 2, one could be deleted or duplicated. Inversions, where the sequence is effectively swapped at a position. And inserts and deletions, where you can have a normal sequence a small amount can be inserted or a small amount can be deleted. So I want to just give a very brief overview of variant classification before we do a deeper dive again. So this is a very generalized singleton analysis workflow. So when we talk about singletons, it basically means the proband or the child that we are looking at. So all sequence variants are added into the funnel, and only those that pass laboratory quality metrics or high confidence variants are allowed through. You might only look at the protein coding variants, and then excluding common variants. So if they're in the general public, they're a lot less likely to be causing the genetic disease at hand. And from there, you're left with a number of variants that you can look at different databases to see if there's any evidence to show that changes in these genes or these variants have been reported before. Other analysis types include trio analysis, and some of you may be aware of this, where the mother, father, and the child is looked at, and this is really helpful because you can look at modes of inheritance. And another analysis type is sort of a paired analysis, where you're looking at differences, and this tends to be more in the tumor space, where you're looking at differences in the tumor from the normal. So an example trio analysis looks a bit like this. So all probands and family variant calls are added, and variant calls are excluded that do not pass the laboratory's quality metrics. Family-based analysis can then occur, and it's using the family structure to focus on relevant positions. Then variants that are classified as likely benign or benign can be excluded, and this is looking at variant classification databases. Variants that are too common in the general population to cause genetic disease can then be excluded, and that's done based looking at allele frequency. Variants with consequences can then be looked at, and excluding presumably non-damaging variants. So we call these synonymous variants, those that don't change the proteins, etc. And then because we have that family analysis, we can filter on inheritance patterns specific to the case. So if we're suspecting an autosomal recessive disease type, we can look at that. Or if we're suspecting an X-linked disease for an affected male proband with unaffected parents, we can look at those as well. And effectively what you're left with after you've done this filtering is a list of candidate variants for clinical review and triaging. Now, the community has been very lucky in the sense that a number of years ago, very good guidelines were published, and rules for combining the criteria to classify sequence variants have been put in place. And effectively what this is, it's a table essentially helping people filter and assign a level to these variants that are being seen. So whether or not it's benign or disease causing as in pathogenic. And when I talk about that, I'm talking about, if it's pathogenic, the variant is documented as clinically significant in multiple publications, or it may overlap with a region with known clinical significance. If it's likely pathogenic, this variant has been described in a single case report, or likely a single case report. Usually has well-defined breakpoints and a gene within the copy number variant that appears relevant to the clinical phenotype. Variants with uncertain clinical significance, it contains genes, but it's unknown if they are dose sensitive, or the data about the copy number variant is contradictory, or there is conflicting evidence in literature about whether or not it's benign or pathogenic. Likely benign, the variant has been described in a few cases, doesn't contain any genes or represent a common polymorphism. And a benign variant is not associated with Mendelian disease in the literature, or it has been documented to represent a common polymorphism or a common change in the general public. So another thing that happens, as well as the variant curation, is something called gene curation. So a clinical whole genome test presents significant challenges for gene curation, and one is at a scale. So looking at the whole genome compared to panel tests, which contain smaller numbers of genes, usually is usually designed to previously well-defined gene-disease associations. Another is that despite having a highly skilled curation team, it is often impossible with one team to have specific expertise for every gene in the genome. Curators need to gain knowledge rapidly on the gene, the mechanisms, and the clinical nuances of the disease before curating a variant. And then there's also the challenge of the turnaround time for the case analysis. These challenges often require a protocol that will work within the given timelines and will provide a framework to guide the curation to ensure consistency and accuracy across whole teams. So things that are looked at is, does variation in this gene cause disease? And before defining the role of a variant in disease, we need to assess the evidence for the association of the gene in which the variant is found with the disease in question. And then how should that potential causative variant be reported? So you've got to look at things like the pathogenicity of the variant, strength of the gene disease association, and the overlap of the disease with the proben's phenotype. So an example gene curation workflow, and this is one that the Illumina Clinical Solutions Laboratory follows, is that there's a collection of evidence, so primary literature or publicly available databases. So a literature search is performed. The identification of the different evidence types occurs, so whether or not it's a curation of genetic evidence or experimental evidence, and then the evidence is assessed. So there's an assignment of points for the collected evidence, and we use a complete summary matrix, and then the calculation of the clinical validity classification, so the strength of that gene to disease association. And from there, it's the scientific and clinical review within the lab. So that's it about how it all works. I want to touch on more now about how it's been utilised in healthcare, the diagnostic utility essentially of whole genome sequencing, etc. So there are several variant classes that can impact genetic disease and a toolbox of testing methodologies that are capable of detecting each class of variant. There are several DNA sequencing testing options that are capable of detecting point mutations represented by the single nucleotide variants and indels category. However, the number of genes targeted by each assay varies greatly from a single gene to a whole coding region of the genome, and whole genome sequencing offers the ability to identify point mutations across all coding and non-coding regions of the genome. There are several assays that can detect copy number variants, but each assay is defined by its resolution for calling a copy number change state. So chromosomal microarrays and whole genome sequencing uniquely offer an ability to assess genome-wide copy number variants. The number of repetitive sequences at certain disease loci can disrupt gene regulation and function. So PCR is standard of care for repeat extension estimation. But there have been recent algorithm developments, so changes in the bioinformatics pipeline that are really demonstrating the ability to estimate repeat expansions from short-read whole genome sequencing data. And as long-read sequencing becomes more prevalent and becomes integrated into healthcare, we'll see that this becomes standard of care for repeat expansion testing. Large structural variants involving the rearrangement of chromosomal material can impact gene regulation by transposing a gene from its typical regulatory environment. These are typically copy neutral events, so no overall imbalance of genetic material, and can be challenging to detect apart from traditional cytogenetic methods. Mitochondrial disorders are an important class of genetic disorders that influence cellular metabolism. Whole genome sequencing is uniquely able to robustly detect each of these variant types, making it the best test to aid with rapid clinical diagnosis of pediatric rare disease. And you can kind of see, whilst the current testing options definitely have a place, there is more call for a singular test that can do all of this testing and therefore decrease the overall time of the testing that's performed. So I don't need to tell you, the current approach to rare disease diagnosis imposes lengthy time to diagnosis and complicates the workflow. So essentially, it's iterative genomic testing, and it creates a really long patient journey to diagnosis. And it's all based on hypothesis testing. So if we have a presentation of a patient and it's suggestive of a specific genetic condition, then a confirmatory assay is generally what's ordered. Now, if there's a diagnosis, great, that's the end of the patient diagnostic odyssey for that particular patient. But if it's not, and a genetic disorder is still suspected, then it moves into this next bucket of testing. So if there are multiple presenting features, dysmorphism, multiple congenital abnormalities, dev delay, or anything else, there are a few different ways that genomic testing can occur. So again, we have our targeted assays, so single gene sequencing, multi-gene sequencing, mitochondrial DNA testing, FISH, so fluorescence in situ hybridization, or repeat expansion by PCR. Or it could be genome-wide assays, so it could be karyotyping, chromosomal microarray, and whole-exome sequencing. But what if this went straight to whole genome sequencing? There's enough to suggest that it can serve as a first-tier test to reduce the patient diagnostic odyssey, and that is something that we are very passionate about at Illumina and even personally. So whole genome sequencing does offer greater diagnostic yield over conventional iterative testing. Now, I was involved in the adoption of chromosomal microarrays about 10 plus years ago into the lab. I remember just my mind's being completely blown at the increase in positive cases we had. So karyotyping has a diagnostic yield of about 5%, and all of a sudden we had a diagnostic yield of about 15%, and that was huge, you know. So instead of 1 in 20, we had more than 1 in 10 cases being reported as having a diagnosis. Now, when you compare that to what's available now with whole genome sequencing, there's an improved diagnostic yield of about 32 to 55% versus the chromosomal microarray. And even whole-exome sequencing improves diagnostic yield by up to 18% when you compare it to a microarray. So this is my last slide, so hopefully I've left enough time for questions. But I just wanted to leave it on this, you know, where whole genome sequencing does offer the advantages over conventional iterative testing, has the improved diagnostic utility. There's a growing body of evidence that whole genome sequencing supports improved diagnostic utility and a faster diagnosis. It's a faster, more efficient workflow. The whole genome sequencing enables robust coverage of the genome and a comprehensive assessment of a diverse range of variant types. And of course, the time and cost savings. Whole genome sequencing can shorten the patient journey with fewer iterative genomic tests required. And that shortening of that diagnostic odyssey is what we constantly strive for, and why we are really passionate about seeing the implementation of whole genome sequencing as standard of care for rare disease in healthcare. So on that note, I'm going to leave it here. I'm sure that there are some questions. So, yeah. Great, hold on.
Yeah, there are some, Renee. Nerida, do you want to ask a question? Asking?
Hi, Renee. My name's Nerida. I have an 11-year-old daughter. We've actually had the whole exome sequence done and the whole genome sequence done. And interestingly enough, they both came up with the same gene. However, it's de novo, and they still haven't proven it as yet. But that took years. So now I sort of understand why these very rare diseases take so long in trying to then analyse what they've actually found. And it's a mitochondrial disease. So sorry, I've forgotten what my question was. So what are the steps when they can't? What are the steps when they can't actually find the other gene, and then what are the steps to then proving that the gene that both the different labs found is actually what the cause of my daughter's conditions are?
Yeah. So, right, basically, right, the first part of the question, sorry, I was already formulating an answer to that second part. So the first part of the question was more about how it's discovered and how they determine it's the same. Is that correct?
Well, it's more a case of they've both come up with the same gene, but because it's a de novo gene, and they're saying it's likely to be pathogenic in its own right, obviously, because she's very seriously affected. It's what are the next steps to actually proving that what they've found is the answer?
So the next steps there, it comes down to making that information available. So it's sharing of that information to databases, and that's something that the Australian Genomics Health Alliance has a flagship for, is the ability for laboratories to share these variants they're finding that are likely pathogenic, or they don't quite know what the variants are. So they could be variants of unknown significance, but they seem likely to be causing the disease state. Then what it takes then, it's a growing body of evidence, and it's something, having worked in a laboratory for years, it's something we did really poorly. So there was multiple reasons why it wasn't happening as well many years ago. It was a real privacy and confidentiality misunderstanding. There was no real defined, I guess, policies around sharing of variants and information within Australia. So a lot of the databases that Australian laboratories use essentially have published data from other countries, and being able to add our laboratory findings to these databases will enable these sort of findings to be upgraded or downgraded depending on what evidence there is. So the variant sharing or database sharing is a definite need, and also publications. So there are a number of different rare disease projects happening around the world. And in fact, we were quite lucky enough at a pre-HTSA workshop that we had Heather present at. We actually had a clinician from a hospital in the US come and talk to us, and they'd actually found a candidate gene or a candidate variant, and they used something called Matchmaker. And it essentially allows them to say, hey, this variant has been, in this gene, or this gene is maybe associated with this disease, but there is a research project happening. So the clinician actually rang the researcher and said, look, I've got this patient, this is what I've found, but there's no evidence, but I am sure it's causative. And what essentially happened then was this patient was recruited into the study, and is now, that paper is used as a body of evidence for further classifications of that variant in new diagnoses. So it's really coming down to that, the importance of information sharing, and it's all de-identified, of course, but it's that ability for laboratories to add to these databases to say this was the phenotype, presenting features, these were the genes that were found, and genes that are likely to have caused this disease. So, there's a change in protein, or there's a change in gene function. So that's the biggest thing. And as sort of sequencing and genomic technology is becoming more embraced, this sharing of variants is actually happening a lot quicker. Australian laboratories are definitely becoming more on board with it. There's a lot more policy being written around it. There's frameworks. And I think there's a lot of interaction with different patient advocacy groups around this, because it's obviously important. It's your child's data that's being shared, etc., to help further diagnoses. So those conversations are happening, but they're happening at a much greater speed now. Does that make sense?
Yeah, and we're part of two research projects. That's why the testing was done. So it's funny that sharing you're saying hasn't been happening, because it certainly happens on Facebook. It's because there's only 7 known other people in the world currently that have the same variant or the same problem with the gene, and we've found all of each other on Facebook. So it's from various countries around the world. Anyway, that's very interesting. Thank you very much, Renee. I'll let others have a chat.
Okay.
I've got so many questions, but I'm sure that there's many people on here that have so many questions as well. So thank you very much.
No, not a problem. I'll just flash up my last slide with my details on it, so that way, if there's any more questions that come up, please feel free to email, or there's my number.
Yeah, sure. I'm not sure if you're attached to. We're part of the mitochondrial disease at the Murdoch Institute, that research project, the whole genome sequence one with John, and then we're part of the Icardia-Gutierrez syndrome in Manchester with Yarrick Crow. So.
Oh, fantastic! They're really great resources. Yeah, great.
Right. Shall we move on to another question? Thanks, Nerida, for yours too. Janine has one. How much does it cost to have private whole genome sequencing done if it can be done publicly?
Yeah, and this is a really tough one. So there are a couple of laboratories in Australia that have accreditation for clinical whole genome sequencing. And I think, you could be looking anywhere for a single whole genome sequencing at about $4,000 to $5,000. And because that includes all of the time involved in the curation and the reporting processes. When we talk about the $1,000 genome, that's effectively to get it to the point of data analysis. Obviously, there's different laboratories around the world, but you're going to find they're all very similar in that respect. But there are differences between paying for an accredited clinical whole genome sequencing test and a research whole genome sequencing test. And it's usually the clinical whole genome sequencing testing that does have added costs, because it's being reported on by clinical geneticists, and it involves the entire pathway, including genetic counselling as well. Does that answer your question? We are, as a company, we work very hard to try and bring this cost of sequencing down as much as possible. So, it's something we strive for. It's one of our pillars. Every year we talk about it, we strive for it, and we work very closely with our sequencing partners to give them the best price possible.
Yeah, thanks. So, answer your question, Janine? Yeah. Kate had a question. I'd love to know how clinicians decide which diagnosis pathway and how do we try and access whole genome sequencing?
So this is something that we work very hard on trying to understand ourselves. It definitely comes down to education. So I actually work quite closely doing what I'm doing tonight with clinicians as well. And so basically explaining to them about the different genomics testing methodologies available, talking about the diagnostic utility, the increased diagnostic yield, etc. A lot of clinicians, when they've gone through medical school, genomics wasn't something that they really covered. The new medical doctors tend to have a bit more of a genomics component to their med schooling. But basically, we have a large number of clinicians come to us directly to ask for help, because we do have those educational resources, and we do go out into the field to talk to clinicians and doctors about this. So for years, there's been like this standard of care when it comes to rare disease, or even just, pediatric disease, where, chromosomal microarray is still considered, the first tier test. So if they're doing a chromosomal microarray, you're probably going to get, your Fragile X or your Prader-Willi or a few other genetic tests that the doctors understand. Now, as Whole-Exome sequencing or whole genome sequencing is coming more and more to the forefront, of clinicians, it's really about getting that message to the paediatricians. And we have some fabulous paediatricians that are just leading the way with requesting genomic testing and whole genome sequencing or whole exome sequencing for their patients. But there is still this sort of educational learning curve that we are facing. So we tend to do a lot in the field as industry partners to educate the clinicians about what genomics testing they can do. And then these very motivated paediatricians are running their own workshops within their group of colleagues to do the same, to bring everyone up to a very similar standard of care.
Megan, did you have a question you wanted to ask Renee?
Yes. Can you hear me properly?
Yeah.
Can you hear me properly?
Yeah.
Yeah. Sorry, I just had jaw surgery, so I'm a bit muffled. I just wanted to ask. My daughter has a gene, MEF2C gene, on the 5q chromosome, which is a deletion, and I've been told that it's very on the same pathway as Rett syndrome and Angelman's and Fragile X. And I was still a bit confused about how some genes could be on similar pathways. If you can sort of briefly explain at all.
Yeah. So there are. When it comes, sorry.
Yeah, sorry. I'm just saying. Yeah, if you can briefly explain, because I know you're probably short for time.
I'm okay. I'm just aware of everyone else. I can stay online as long as you need me to. So when different genes can disrupt the same pathways. So when it comes to the way that genes tend to interact with each other, it can start to look a bit like a spider web when we look at the pathways and how they interact. So what can happen is that one singular gene that disrupts that pathway can interrupt the entire pathway. And so because of that, it can lead to very similar sort of phenotypes or presenting symptoms. Now, different genes can also encode for very similar features, and a disruption of those features can lead to, again, very similar presentations. So when your daughter may have presented, it was probably the doctor looked at her and went, oh, it could be this, it could be that, because these features of these different diseases are very common to each of these diseases. So it could be that it's within the same pathway, or just a disruption in these genes can lead to very similar features. And the way that the doctor has tried to explain it to you is that it's similar to these other disease types that are more commonly known, maybe.
Yep. Okay. Yeah, because that's the first thing they said when I first got diagnosed with the geneticist. He said it was on the same pathway as Rett syndrome, and I said, oh, okay. And it sort of left me really confused. But yeah, thank you.
That's okay.
Thanks for explaining that. Makes more sense.
There's one other question from another header. There's 7,000 known diseases as far as I understand. Do you know how many more are being discovered per year? Just curious the rate of progress here.
Oh, so am I. That's a brilliant question. How about I do some research and get back to the group, because that's really piqued my interest as well.
Yeah,
so
I'd be interested.
Yeah, look, I'm actually just going to take that as a copy paste, so bear with me. I think that's a fascinating question. So let me get back to you on that one, because I'm sure there is some literature that we can find on that. I do know that genetic disease research is just one of the biggest areas. It's right up there with oncology, and that is led a lot by pharma. But the genetic disease research into rare disease and just genes in general is a massive part of what we help support. So I am sure I can find some numbers on that for you. But fabulous question, thank you.
One more from Nerida. It's interesting that a problem in protein seems to be the cause of many diseases. My daughter has a problem in the CLPB gene, which is a protein that feeds the mitochondrial.
Yeah, so basically the DNA through that transcription and translation process does lead to proteins, and it's the proteins that drive a lot of these cellular processes. So it's a lot easier for us to look at DNA because it's more readily available. We're seeing a lot of work in RNA sequencing, and I think we're going to see that happening in the clinic more and more. And there's, of course, protein studies as well, so proteomic studies. And in fact, one of the groups I was involved with at one of my previous labs had a protein component to it because it was deemed, super important. We understand the genomics and the protein on the proteomics as well.
Interesting. Same with Soto's narrator, the NSD1 produces a change in the protein and that causes the issues. Interesting. Renee, I've got a question. You said in your talk that you can use blood samples or saliva samples. Is one better than the other?
Not really. So basically, they both allow for high quality DNA. And I think what we're seeing, blood was just always deemed more readily accessible. It was what was already being taken at the time for testing. So when genetic testing is usually requested, there are other things requested. You might see things on your form like full blood count, biochemistry markers, etc. So another tube of blood for genetic testing seems appropriate. Now, saliva or buccal swabs, where they do the cheek swab, is becoming more and more popular because obviously there are definitely issues sometimes getting needles into arms and extracting blood from patients. So there's been a huge rise in the number of saliva and buccal swabs for DNA. And in fact, it's so popular and the need is so great that we've developed and just recently released library preparation kits that can do both blood and saliva as direct input. Now, there are other different types of tissues that can be collected for DNA. So there's muscle biopsies, which are usually better for when you're looking at mitochondrial disease. But a lot of the time, that's usually a punch biopsy and it can be quite painful. So you want to be really sure that it's a mitochondrial disease before you want to do something that invasive. And right the way through to stool samples if you're doing sort of more microbiology applications. So pretty much anything you can extract DNA from you can use, but when it comes to rare disease, it tends to be blood and saliva.
Okay, thank you. There's one more question. Are you able to explain how you know whether a condition or presentation is caused by one pathogenic genetic variant or whether there could be a second issue? Our child was diagnosed with one genetic condition, Fragile X, but we're suspecting there is something else.
And that's a fascinating area of research because it's quite often a common theory amongst people that, finding one cause does not mean it's the only cause. We had a saying in cytogenetics, don't miss the obvious for the obvious. So just because you found, say, a Down syndrome patient, don't miss the obvious 6q deletion or something like that that's also happening. So when it comes to this iterative testing, what we do find is that a singular diagnosis is made and then that's the end of the diagnostic odyssey or the diagnostic journey, but there is still something else happening. So this is where further genomic sequencing, so whether or not that's Whole-Exome or whole genome sequencing could probably be really useful here because there can be almost a confirmation of the Fragile X, which you understand about, but there's also that ability to find what else could be happening. And it's quite a common theory that in rare disease, it could be multiple things happening, and that's what makes the presentation so severe, rather than just a singular gene disorder. It could be a few genes that have been affected. So definitely keep advocating for more genetic testing if that's what you suspect.
Thanks. Yep, good question. Does anyone else have any more questions for Renee?
I do, Heather. It's Nerida here.
Yep, go for it.
Renee, so I'm the complex I'm one of the complex ones also on this call. So, so my daughter is the one that I spoke about earlier. So she's got a very rare blood disease, and she also has a very rare brain disease. So what they're looking for is the overall cause of those combined diseases, if you like.
Yeah.
So they did do because they've never been able to rule out mitochondria, so that's where they've sort of been focusing on of late. And so they did the punch test, the skin punch, because she has a rare blood disease. I denied the liver and muscle biopsy because the risk of infection for her is way too high to not have a definitive answer, which I understand. You know, these tests can come back negative, positive, or we still don't know. So the skin seemed to be the least invasive in our instance. So because they can only find one gene, and this was a while ago, it's been probably a year and a half since I've last had any correspondence. So forgive me for not remembering all of the scientific terms anymore. But so they were doing obviously some tests. They were looking for the other gene. So they said that they had to do some other testing, some fibra, I can't remember the name. So how would they have not been able to find the other gene? I don't really understand. If all the genes are there and they've found one that's got a fault in it, I don't really understand how they can't find the other one.
Yeah. So, and that's the crux of a lot of what we talk about when it comes to that gene curation. So, what tends to happen is, even though a whole genome sequencing test is done, that filtering funnel where one of the filters can be associated with this disease, it almost precludes the ability for, I guess, discovery testing to occur, because a lot of labs really are focusing on evidence-based diagnosis or evidence-based medicine. I suspect what the testing was fibroblasts, which are the skin cells.
That's it. Yeah.
So,
that's it.
Yeah.
Yep.
So what would have happened there? They would have cultured the skin cells to further extract DNA from that. So from there, they can do further testing or store that DNA for future requests, etc. So if the entire, so if there was exome or genome sequencing performed, what could be requested is something called a reanalysis, where in 18 months, there has been a huge number of different breakthroughs in different rare diseases, and something else might pop out because that diagnosis was relevant to the genes that were understood at the time. So this is where, even in the exome sequencing MBS item numbers, reanalysis is hopefully going to be one of the item codes to allow for situations like this, where there was a diagnosis potentially made at the time, but the second hit or the second gene wasn't identified. So reanalysis with the knowledge that we have today could really make that difference.
Yeah, I'm sure that will be happening in the background, because, as I said, it's all part of a research project, and I was asked to give permission for my daughter's case to be published. Yeah. So,
Great.
Because if they do prove it, it's a gene that hasn't been proven to be pathogenic in its own right as yet, and if they do prove it, then obviously it's a bit, because I understand there's not that many genes that are pathogenic in their own right. Is that correct?
No.
There's very few?
That's true. So there are multiple genes that changes can be associated with disease, but all of that needs to be sort of tested or functionally tested. And I presume this is what's happening with you also, like the fibroblasts, etc., is that they're going to be almost stress testing them.
Yes, functional studies. That's exactly the terms that they were using. Yep. It's all coming back to me now.
So, I mean, this is where it's patient families such as yourselves. So, you are really driving the forward leaders on this, new genomics frontier, if you like. you've been a part of the diagnostic odyssey and the patient genetic journey for so long, and now there's this new technology that can potentially help and is helping, but because you're at the front end of the utilization of genomics testing, you're going to be helping define and diagnostic testing for the future, and also the ability for you to be sharing that data so it can be published, the variant shared on databases. That's what's going to decrease the diagnostic odyssey for other people in your shoes. So I understand it can be frustrating because you've been living this for a really long time and technology has changed and it's revolutionized healthcare. But it is people such as yourselves that are consenting for this data sharing that's going to really make a difference for families in the future.
And that's why, that's why we're doing it, because at the end of the day, I guess we gave up quite a long time ago about finding an overall diagnosis. So, but of course, as soon as they start to talk about proteins and things like that, I then start to ask questions like stem cell therapy and all of that sort of stuff. Because if it's a protein, then surely that means that, there can be some stem cell therapy out there. But of course, my questions were way too ahead of the game in that regard. So.
Oh, look, and it's something, I think that's something that we're seeing as people in the field, is that it's the patient families, it's the advocates that are really driving a lot of this themselves. So it's been really interesting to partner and work a lot more closely with Heather and other sort of patient advocacy groups about how all of this works, because I think we're learning a heck of a lot from you guys, because you share information. Like you said, you've been connecting with other families on Facebook. It's stuff that we don't do. We're sort of limited in what we can share and what we can look at, but you guys are making that connection, sharing that information, talking about what medication works for your children. That's fascinating. So.
Exactly, that's exactly what we're doing. It's funny because I'll go back to the hospital and have sort of said, we found another CLPB, and we're all different, of course. All the kids are different, and most of the kids that I've found are not de novo, and it's interesting. They're all different, affected differently, and they all have neutropenia, which is the blood disease, but they all have it in various forms. And my daughter has it in the most serious form in the group. So, and most of them are in the Eastern European countries, actually. And there's a clinician over there who is part of the Facebook group. So, she encouraged one of the other de novo parents to join the group. So it's great. And exactly about what sort of, not necessarily even medications, but supplements. And because when mitochondrial, I'm sure we've all had to try the mitochondrial cocktail for our kids, and to try and work out whether or not, and interesting that thiamine's on that list of supplements when we're all trying the mitochondrial cocktail. So it's it's really interesting. It's so fascinating. It's just a shame that we have to live it, but it's very fascinating. Sorry, I'll let others speak if others have got questions.
I'm just conscious of the time, and I think Renee's put her details up, so perhaps if anyone's got any more questions, they can email or phone Renee. Obviously not tonight, being a Sunday night, but in work hours. So thank you, Renee. Really fascinating talk, and I'm sure you'll get some emails. And I look forward to talking to you some other time and putting your presentation up online.
No, it's been a pleasure. Thank you.